Hi, my name is Julia Sciuchetti, and I'm here to present my research project, Molecular Diversity of the West Nile virus in Montana. So some background on this. West Nile virus has been present in the United States since 1999. It was first detected in Montana in 2002. As of September 4th, 2019, there have been 326 cases of West Nile virus in humans, and 45 states have reported infection in birds, mosquitoes, or horses. So the virus itself is a single-stranded positive sense RNA virus, and the disease has two classifications, neuroinvasive and non-neuroinvasive. 80% of people that are infected have no symptoms, but for the 20% that do, they can be very severe. And for those that show symptoms, it's most severe in humans and horses. So as you can see on my map here, localized environmental conditions can affect viral persistence. So at Carroll, we have predictive models that can help us predict where equine vaccination efforts should go. The Center for Disease Control and Prevention tracks disease and virus progression through surveillance programs like that which we have here at Carroll. So the West Nile virus has a very high mutation rate that makes it difficult to create a human vaccine. So the rate is higher than the influenza or than the measles virus, but lower than the influenza virus. So, which makes it difficult to make a vaccine, like how the flu, you need a new vaccine every year. So the envelope gene is chosen for this study because of its high variability and information that can be comparable to whole genome studies. The sequence length for that is 1,575 nucleotides, which is about 14% of the whole genome. And the gene product for that is a 53 kilodalton protein that's a target for virus neutralizing antibodies that could potentially be used in vaccine efforts in the future. So like I said, the overall goal of this study is to compare envelope gene variation of West Nile virus found within geographically distinct mosquito populations across the state of Montana and contribute to the larger project of creating a risk assessment map for the state of Montana, which is shown in the center there. So for my methods, from June to August, from 2009 to 2019, mosquitoes were trapped using carbon dioxide baited traps and then sorted to species. They were put into pools of 50 or less and labeled with their associated location. And then they were put into the and then they followed the protocol outlined by Black and Dutal of 2001, and positive detection was done following the protocol outlined in Lanciotti et al. 2000 in order to test for positive virus in the mosquitoes. So part of that was run here at Carroll College, and the state of Montana Health and Human Services also tested the mosquitoes that we found. So we did then RNA extraction and polymerase chain reaction here at Carroll. And then, let's see, I did in my project two overlapping envelope genes following the protocol of Bertolotti et al. 2007 with the QIAGEN one-step RT-PCR with 2.5 milliliter RNA template. And that was done in either 25 or 15 milliliter volumes with the BioRad thermocycler, and then it was analyzed using gel electrophoresis on 1% agarose gels at 100 volts for 30 minutes, and then visualized under trans-UV light. So for results, you can see for summer 2019, we had 423 sample pools, and there were 7 West Nile virus positive samples. So that represented 4 counties, so Valley, Blaine, Sheridan, and Yellowstone. So that's represented by the green dots here on the map. The black dots show where West Nile virus has been found in the past. And then these yellow triangles show where there are testing efforts for the West Nile virus up here too. Um, so as you can see in my gel below, um, after 18 trials of gels, I did get banding for all of the samples shown. You can see uh, this was the correct length or the desired length that I was looking for, which was 829 base pairs for the first primer set, which was 871 and 1700 base pairs. And then for the second primer set, I was looking for this this banding pattern to the right, which was the uh, uh, 1071 base pairs, and that was the 2666 and 1595 base pair primer set. So after it was shown that we got amplification of the correct length that we were looking for, this was sent for sequencing to Macrogen USA. But unfortunately, the results are inconclusive and unable to be compared after sequencing. So then for my conclusion, my hypothesis was neither accepted nor rejected because the sequencing and comparison efforts were inconclusive. I had a lot of issues with the trans UV viewing camera, which allowed for why I needed that many trials to get gel visualization. There was also issues with the protocol with the RT-PCR amplification actually amplifying the correct length. As you can see in these gels, there's a lot of faint banding below the bands of the desired length, which are probably primer dimers, but also just random amplification that was undesired and caused a lot of noise in the sequencing efforts and made it difficult to actually compare anything. And then also the sample size was fairly low with only 7 pools testing positive this summer. So I was hoping to aid in the overall project. So since that of making the prediction map, so since there was one pool or one location of 3 pools actually is this dot that was further south versus the other 3 green dots are up north in Montana. So we're hoping to kind of compare geographically north versus south. And then eventually it would have been interesting to compare to other states. So Bertolotti et al. In 2007 in Illinois found that geographic subdivisioning had little effect on sequence because of the increase in mutation rate. Like because the mutation rate was so high, there was just not a lot of differences between the West Nile virus sequences in Illinois. But Ebel et al. In 2004 found that populations were homogeneous in New York, and he believed that this was, or they believed that this was due to a new genotype that was introduced in 2002 that just kind of spread and outcompeted the other sequences. They believe that there was more diversity before the introduction of that novel genotype, but because of its rapid and facilitated transmission, and it was believed to be under immune selection, that it was able to just really be homogeneous. So it would be interesting to see if Montana as a state was more like New York or would be more like Illinois. So it's also important to keep up on this because of the antigenic importance of making efforts with the envelope gene toward a human West Nile virus vaccine. So understanding the evolution diversification can allow for better predictions of outbreaks as well, which allows for improved public health measures for both humans and horses, which is what the overall project here is at Carroll. As you can see in the West Nile virus risk map that Dr. Glowinka and Dr. Hokett have been working so hard to continually refine. So in the future, research could be interesting of east and west of the Rocky Mountains, since those are a much bigger geographical barrier than just north and south. Uh, Also, whole genome studies would be potentially more informative than just one gene. And then finally, this research definitely needs more replicates and samples, and hopefully my work has helped refine the protocol for future students and studies. Overall, I would like to thank Dr. Jennifer Glowinka and Dr. Grant Hokett for their continued support and help and guidance. And I would like to thank all that worked on this project in previous years. Um, and I would like to thank the National Institute of General Medicine Sciences of National Institutes of Health under our award number that I've got down there at the bottom. Yeah. Thank you for watching.